Hello friends, welcome back to my channel. Hope you all are doing well. In this video, we are going to look at the cell envelope in prokaryotes. Yeah, so you all must be thinking, what is the cell envelope? As we all know, the prokaryotes are unicellular. Hence, for the survival, the cell should definitely be blessed with some stronger covering. This protective covering is called cell envelope. It's the combination of three layers, the glycocalyx, the cell wall, and the plasma membrane. Basically, it provides structural integrity to cell and protects it from internal turgor pressure caused by higher concentration of macromolecules like proteins or some ions inside the cell. So, in this diagram, the three layer is clearly visible. The outermost, the middle and the innermost. The outer one is called glycocalyx, the middle one is called cell wall, and the innermost one is called cell membrane or plasma membrane. They formed a cell envelope. Let's we take these three layers one by one to understand more. So, the first outermost layer is glycocalyx. It is made up of polysaccharides and some proteins. It occurs in two forms. First is slime layer and the second one is in the capsule form. If the constructed unit glycocalyx arranged in loose sheet-like structure, like you can see over here, it is then called slime layer. If the glycocalyx having compact and tightly arrangement, then it is called a capsule, which is much thicker and tough covering. So functionally, glycocalyx or the outermost covering helps the bacteria to adhere to the surfaces. Now the second layer is cell wall. Just inside the glycocalyx there is a cell wall which is composed of peptidoglycan in eubacteria and pseudopeptidoglycan in archaebacteria. So if someone asked about the difference between bacteria and archaebacteria, you can easily answer the difference is in their cell wall composition. Okay. Now, as we know, bacteria are classified in gram-positive and gram-negative. This solely depends on the composition of cell wall and does not depend upon the glycocalyx or cell membrane. Glycocalyx, the cell wall of different bacteria show different types of reactions with gram stain. So, how we will distinguish gram-positive bacteria from gram-negative bacteria in a given sample of bacteria? For this, we have to perform gram staining technique. So, let's go for it. Now, what is the gram staining procedure? It involves two staining processes. The first stain is crystal violet, which gives violet or purple color to the cell, and the second stain, which is counter stain, gives pink color. So this is the pictorial representation of gram stain technique. Where is this? Here. In the first step, we have to stain the bacterial sample with crystal violet. Okay, this is the first step. We have to stain the bacterial sample and all bacteria become violet or purple like this. They all become violet or purple in color. In the second step, we have to stain this with iodine, which form complex with crystal violet and retain the violet or purple color. They retain the same color. So now in the third step, decolorized the whole sample with alcohol. Decolorization is done with the help of alcohol. Some bacterial cell decolorizes and lose the purple or violet color. They fail to retain the crystal violet color and become white. In the last step, add safranin, which acts as a counter stain to the colorless cell that were formed in the step of decolorization. Now, in the bacterial sample, some are violet purple in color, which retain crystal violet stain are gram-positive bacterial cell, and those which are pink in color, counter stained by safranin, are gram-negative bacterial cell. This is because the peptidoglycan layer is very thick in gram-positive bacteria as gram-positive bacteria. Cell wall with thick peptidoglycan layer which help in retaining the crystal violet stain and appears blue in gram staining technique that we see later. Teichoic acids and lipoteichoic acids also embedded in cell wall. The gram-negative bacteria. The cell wall is thin, very thin, having peptidoglycan layer which is very thin as compared to the gram-positive bacteria and which is unable to retain the crystal violet stain when decolorized with alcohol during gram staining technique. Beside thin layer of peptidoglycan, it also have an outer membrane composed of lipopolysaccharides and phospholipids and also a periplasmic space. Here. So this is the picture showing the cell envelope of a bacterial cell. The cell wall is so much thick in gram-positive bacteria, which is made up of peptidoglycan, and in gram-negative bacteria, the cell wall you can see is very thin. There is also the existence of an outer membrane in gram-negative cell. This is the outer membrane which is structurally very similar to our plasma membrane. Let's we take some other picture from which you can clearly understood the difference. So this is the another picture. In gram-positive bacteria, the outer covering is very thick. The outer covering that is made up of peptidoglycan layer is very thick as compared to the gram-negative, and in gram-negative there is a outer membrane. This is the outer membrane inside which these two in blue colors are these two in blue. These are periplasmic space, and the inner one which is green in color is the plasma membrane. So basically the difference between gram positive and the gram negative is solely depends on the structure of cell wall. If the cell wall is thick, then it is called gram positive bacteria, and if the cell wall is thin, then it is called gram negative bacteria. So let me tell you one more category of bacteria. These are Mycobacteria. Here cell envelopes is different from gram negative and gram positive bacteria. It lacks an outer membrane, hence there is no outer membrane present in Mycobacteria, which is present in gram negative but has a cell composed of peptidoglycan, arabinogalactose and mycolic acid. Now, the innermost layer is plasma membrane. This is the plasma membrane which separates the cytoplasm or interior of the cell to the exterior surrounding, which is called extracellular fluid. The plasma membrane is composed mainly of phospholipids and embedded proteins. It allows movements of ions that you can see over here. There is a transport of molecules taking place with the help of these embedded proteins. It is very selective in nature. The structure that you can see over here, this is the phospholipid having hydrophilic head. Hydrophilic means loving water. Hydrophilic head and the hydrophilic tail. These are fatty acids tail. Heads pointing outside as they are hydrophilic and fatty tail pointing inwards as they are hydrophobic in nature, which hates water, and thus forming lipid bilayer. That's why it is called lipid bilayer. So we can say this is the innermost layer of cell envelopes of prokaryotes, is structurally composed of phospholipids bilayer in which protein molecules are embedded. Most of the functions are performed by embedded proteins. It serves the site for photosynthesis, respiration, cell wall formation, and lipid synthesis. It acts as a receptor and antigen which helps bacteria to respond to external chemical stimuli. The semi-permeability provides selective choosing of molecules to pass through it. Now, next, why these cells are blessed with three layers, that is cell envelope. We all know we used envelope to protect our things which we keep in the envelope. Just like that, these unicellular organisms protects themselves from rude outer environment. These three conjugative layers perform their specialized functions. First one is Glycocalyx, which help in attachment of surfaces, the cell wall, which maintain the shapes and protect it from bursting or collapsing, and the plasma membrane, which regulates the transport of molecules in and out of the cell.